Good morning, all. This is Professor Arun Ingre from School of Life Sciences, Kavitri Bainabai Chaudhary, North Maharashtra University, Jalgaon. Students, today we will discuss about the bioinformatics, a brief overview of the usage in the plant sciences. Now, let us discuss with the bioinformatics. What is bioinformatics? Bioinformatics is the application of the information technology to storage, management, and analysis of the biological information. As such, there is no general definition about the bioinformatics. So there are several definitions made by the different scientists regarding the bioinformatics. But one thing is common. So storage, management, analysis, and retrieval of the biological data, and that is facilitated by the use of computer. This is comes under the bioinformatics. So what can be done using the bioinformatics? Sequence analysis. First of all, the sequence analysis is the major part of the bioinformatics. So uh, many scientists working on the protein sequence, DNA sequence, and many other biomolecules. So the huge data is produced by the scientists, and that data, there is a dire need of the use of that data in a sequential manner. So that sequential manner application is the storage, management, and retrieval of that data. So in sequence analysis, the protein sequence and DNA sequence are used. So the DNA sequence can be done, sequence analysis can be done in pairwise alignment. Protein sequence can also be done by pairwise alignment. Means pairwise alignment and multiple sequence alignment can be done by using this sequence analysis tools by using the bioinformatics. So geneticists and molecular biologists analyze the genome sequence information to understand disease processes. Secondly, the molecular modeling is one of the very important area of the bioinformatics. Those people who are working in the lab on a protein, they got the protein structure from the NMR, X-ray diffraction, and XRD. So this exocrystallography. So these are the techniques in wet lab work we can do for the determination of the protein structure, and that protein structure is deposited in the protein data bank. If those people are not having their protein structure, they can model the protein structure from their sequence. So the crystallographer or biochemist use this structural information of the molecular modeling for the drug design and for the using the computer aided tools. Phylogeny analysis Phylogeny analysis and evolution study can be done by using the bioinformatics. Genetics obtain the information about the evolution of organism by taking and simulating in the gene sequences. So phylogeny means we can say there is a there is a use of the we can say typically graphical and representation of the evolutionary relationship among three or more genes or organisms. Phylogeny tree are made by the node and branches. Every node represents the taxonomic unit and rooted trees and unrooted trees. So this output of the phylogeny tree is related to the we can say the common ancestor can be identified from this phylogenetic analysis. Ecology and population studies also can be done by using the bioinformatics. So bioinformatics is used to handle the large amount of data obtained in the population studies. Similarly, the medical informatics also use the bioinformatics techniques for the personalized medicine. So bioinformatics is having the key areas: sequence analysis, structural bioinformatics, and databases. So you can see there is a homology searches. If you have the protein sequence, so you can go for the homology searches. Means you can identify your sequence which is already studied or not. Similarly, structural bioinformatics you can maintain, produce, or develop or determine the protein structure by using the bioinformatics. Another area is the databases. Means huge data is produced every day by different scientists worldwide, and maintenance of that data is important. So databases are playing a key role in the maintaining the data. Why there is need of bioinformatist? So there is a need of bioinformatics because the number of the entries in the database of the gene sequence is increasing exponentially, as well as protein sequence also increasing sequentially, exponentially, or logarithmically. Bioinformaticians are needed to understand and use of this information. You can see here the logarithmic growth of the gene bank. Gene bank deposited deposit the protein sequence, gene sequences, and here you can see there is a large amount of data is deposited in the gene. So now moving towards the databases, biological databases. There are different type of the biological databases found in uh, the internet. We can say the worldwide, the biological databases are a library of biological sciences collected from scientific experiment. Means the data which is collected from scientific experiment and also published literature, just like the scientific experiment of the high throughput experiments. So biological data plays a central role in the bioinformatics. They offer scientists the opportunity to access the wide variety of the biological relevant data, including the genomic sequence of the increasing broad range of microorganisms. And this type of databases are found online. So different scientists develop this type of the databases. So these databases are categorized into three: primary database, secondary database, and tertiary database. In primary databases, the sequences are deposited. Sequences of the protein or DNA are deposited in the primary databases. Secondary database is related to the annotation. Annotation means the information related to the sequence. Tertiary, the database is provides you information regarding the structure of the protein. Again, there are two different type of the databases: protein database and DNA database. Gene bank, DDBJ, and EMBL are the example of the DNA database. PIR, SwissProt, and MIPS are the example of the protein databases. So from here we can get the information related to protein sequence and DNA sequences. For that, NCBI is the very important source for the getting the information related to the biology. So National Center for Biotechnology Information advances science and health by providing access to the biomedical and genomic information. This NCPI will provide you every aspect of the biological information. NCPI houses a series of databases relevant to the biotechnology and biomedicine and is an important source for the bioinformatics tools and services. On NCPI, while moving towards the NCPI, there is one page that is entries. From entries page, you can get protein sequences and DNA sequences. The protein sequences are retrieved from the PIR Swiss for databases, and protein structures can be retrieved from the PDB database. Similarly, the DNA sequences can be retrieved, can be collected from the EMBL, DDBJ, and GenBank. So entries will provide you a direct access, just like a single window. So through NCBI entries portal, you can get all information at a glance. So similarly, there is a SRS and SRS single sequence retrieval system available online. If you are interested in getting the publication related information, PubMed and Medline is available on the NCBI site. So moving towards the analysis purpose, phylogeny analysis of the protein sequences. Analysis of the protein sequences is studied in through this bioinformatics software. Analysis of the protein sequences can be takes place in two ways: the pairwise alignment sequence and we can say the multiple sequence alignment. Pairwise sequence alignment is used to identify region of similarity that may indicate the functional, structural, and evolutionary relationship between the two biological sequences, just like a protein and DNA. Now, the sequence alignment is a way of arranging the protein and DNA sequence to identify regions of similarity that may be consequence of evolutionary relationship between the sequences. So multiple sequence alignment is the generally the alignment of three or more biological sequences, for example, protein or DNA of similar length. From the output, homology can be inferred and evolutionary relation between the sequences studied. Third, protein structure prediction. Protein structure can be predicted by using the bioinformatics tools. Similarly, the prediction of transmembrane region can be also predicted by using the uh, bioinformatics tools. For transmembrane regions, we can say these transmembrane regions are found in the protein. That proteins are anchored in a transmembrane region. So how many helix are encoded in a transformed region can be generated, can be predicted by using these tools, by using this bioinformatics. Prediction of secondary structure also can be done by using the bioinformatics. Secondary structure consisting the beta plates and alpha helix information, such as also the coil, turns, strands information available in the secondary structure. So this can be predicted by using bioinformatics. Prediction of conserved domain also takes place by using the bioinformatics tools. Determination of the coil region, prediction of MSC class 2 binding sites. Molecular modeling studies can also be done by using the bioinformatics softwares. So the comparison, first example of the use of the bioinformatics tools is the comparison of the sequences. So pairwise alignment, sequence alignment can be done by using the BLAST. BLAST P, BLAST N, and BLAST N BLAST P are the tools that can be used for the sequence pairwise alignment of the sequences. BLAST is a basic local alignment search tools. So if you are interested in aligning the two sequences of protein, you can use a BLAST P. If you are interested in doing the nucleotide sequences, you can use a BLAST N. Similarly, if you are interested in aligning the two sequences that are that are related to the protein sequences with the DNA sequence, you can use the N BLAST P. Similarly, the multiple sequence alignment can be done by using the clustral W and clustral R. If you are interested in doing the work on a protein, that time you need to use the clustral W. If you are interested in doing the work on the DNA sequence or nucleotide sequence, you can use the clustral W. So another interfaces are available as a user interfaces on the internet that can be used for the different type of the purposes. So BioEdit, Biological Workbench, and CLC Workbench are the user interface that can be useful for the different type of the work on the internet. So these three different type of the work interfaces are available, workbenches are available freely for the academic purpose only. If you are interested in doing commercial work, you need to pay for that, or you need to register for as a commercial user. Now, this is about the NCBI web page. So we discussed earlier that is the NCBI is providing a we can say the single window for the every type of work. Here you can use if you are interested in getting the protein sequence, you can just search put a search box protein and keep keywords as a protein. So after putting the keywords, you will get a number of hits. That hits on after clicking on the hits, you can get a specific type of the information of the protein sequences from the NCBI protein. Now, if you are interested in doing work on the data blast or search of the protein, so database blast is available for the searching similarity between the query sequence and the subject sequence. The query sequence is a sequence of your sequence and the subject sequence of the databases. So you can compare the two sequence and similarity search between the your own sequence or your sequence of your interest and the sequence in the database. So sequence in the database can use for the alignment of the protein, and this alignment shows by blast by color key alignment. Here you can see the red line indicates the highest matching takes place found in the query sequence and the sequence of the database. So on the basis of value, we can get the information which sequence is mostly or highly matched. So multiple sequence alignment is another tool for the analysis of the more than three sequences. More than multiple sequences can be aligned in a software, and you will get a different type of information from the multiple sequence alignment. Multiple sequence alignment is generally done for the phylogeny analysis. So you can produce a phylogeny tree by using the multiple sequence alignment of the different organism. Through this, we can get a information regarding the conserved sequence. Here you can see the star or asterisks are provided below the region of the sequence that can be shown, that is important, that indicates that this sequence is conserved sequence. The sequence is, came from the ancestor. We can say the common ancestor are there in between this all sequences or organisms. Now, phylogenetic tree can be also produced by using the analysis of these sequences. So, clustral W can be used for the development of the phylogenetic tree or construction of the phylogenetic tree for the protein, and for the clustral X for the DNA sequence. Otherwise, you can also use construct the phylogenetic tree using the phylic package that is also available online. This phylogeny tree provides you information between the distance between the two organisms, and there are two type of the phylogenetic tree. One is a rooted and another is a unrooted tree. Now, the structural information. The protein structure can be predicted by using different software. Here is the we can say the at a glance information about the how protein is converted, sequence is converted into protein structure. Primary sequence is called as a primary structure. Linear sequence is called as a primary structure, and it is converted into alpha helix and beta plated sheets through the local interaction that can be called as a secondary structure. This will, if the folding of this interaction of this helix, alpha helix, beta plated sheets, coil sheets takes place, that is called as global interaction. That will be responsible for production of the development of the 3D structure or tertiary structure of the protein. So this structural information is required for the means from in sequence we can get a structure, from structure we can get a functional information regarding the. Now the structural in bioinformatics, you can see the structural bioinformatics is responsible for development or prediction of the secondary structure or 3D structure. So secondary structure can be predicted by using the software. Homology modeling, 3D and ab initio 3D structure can be predicted by using the bioinformatics software. Analysis of the 3D structure is can be done by using the different type of the software. So the structural comparison can be done by using the during the analysis. The prediction of the functional sites from the structure can be predicted, produced. Molecular mechanics and molecular dynamics can be studied. Prediction of molecular interaction and docking studies can be done by using the bioinformatics software. Secondary structure. If you are interested in getting the secondary structure information from the sequence, if you have the sequence of your protein or new protein, you can predict the secondary structure from your protein. If you are unable to, if you don't have the protein sequence of your own, so you can retrieve the sequence from the database of the protein, and that can be used for the secondary structure prediction. So there are several softwares available online that can be used for the prediction of the secondary structure. JPRED is the one example. SOPMA is another. GORE is another prediction software. Simple, we can say these softwares are very user friendly. You can directly paste your sequence in the search box and make prediction. After clicking on the make prediction, you will get a result of the secondary. Secondary structure produced in the form of the in this form means from alignment there is algorithm used neural network and alignment that produces the secondary structure information. Here coil, helix, strands information can be produced by using this secondary structure prediction. So how many strands are available, how many beta plated sheets are present in the protein structure, how many helix are present in the secondary structure can be predicted by using this software. Similarly, the 3D structure prediction can be done by using the bioinformatics tools. Ab initio, there are different type of the methods for the prediction of the 3D structure. Ab initio structure prediction, template-based structure prediction can be used for the prediction of the 3D structure. From query sequence, if you have the protein sequence, you can use your query sequence, protein sequence as a query sequence, and it will the 3D prediction server will use the protein data bank for the template for the modeling the 3D structure. So this template structure can be used, and then that will be aligned with the your query sequence, and then there is a production of the prediction of the 3D structure. So molecular modeling, it is also called the molecular modeling, homology modeling and comparative modeling. So the two types of the modeling is there. Homology modeling means the sequence can be searched, similarity, similarity search. Through the similarity search, the model can be developed. Comparative modeling is done by using the structure which is available in a PDB data bank. So through the comparing with the template, comparing with the structure which is available in a PDB data bank, and the query sequence and template responsible for product development of the molecular model. So there are several softwares which are available on the net that are free. Modeler is the one of the very best molecular modeling software. SysPDVR is also can be used for the molecular modeling. Similarly, GeneThreader and MolMol can be used for the molecular. So after once the molecular model is predicted, that can be visualized by using the different type of molecular visualization tool. RASMOL, CN3D, JMOL, PyMOL, and JMOL like software can be used for the molecular visualization of the 3D structure. Similarly, CPH model is also used for the tertiary structure prediction. Means this model is also used for the development. Here you can see the PDB, protein data bank. Here the protein data bank is freely available for everyone. The people or scientists working in the field of protein, NMR and crystallography structures are deposited in the PDB, and that is available for the prediction of the 3D structure also. If you are working on the protein structure, if you determine the protein structure by using the wet lab technique, that is NMR or X-ray crystallography or X-ray XRT, so you can deposit your protein sequence in the PDB database. Similarly, there is a PubMed database available that is responsible for providing you a literature or citation or the paper published already that can be provided by the through NCBI portal, and that publication is related to the only uh, biology and medicine. So THMM is the we can say this is a transmembrane helix prediction software. So transmembrane helix. So in case of the protein, there are the protein is embedded in the transmembrane region. So from this you can get information how many helix are present in the transmembrane region. This transmembrane THMM, TMHMM, the software that can be predicted, that can be used for the prediction of the transmembrane region. The number of the helix is present in the transmembrane region. You can also use some softwares for the active site prediction. So in protein structure, there are some packets, some cavities are present. That cavities and packets are used for the binding of the drugs. So that site is called as a active site. That active sites are used for the designing of drugs. PCR primer can be designed by using the bioinformatics software. Similarly, restriction mapping. Gene can be analyzed to detect the gene sequence that can be cleared with the restriction enzyme. This type of the work we can do on using the bioinformatics. Similarly, the RNA structure prediction, structure feature of the RNA can be predicted by using the uh, now moving towards the role of the bioinformatics in the uh, role of the bioinformatics related to the plant genome analysis. So bioinformatics plays an important, we can say essential role in today's plant science. As the amount of data grows exponentially, there is a parallel growth in the demand for tools and method in the data management, visualization, integration, analysis, and modeling, as well as the prediction. So there are several softwares we can use for the prediction of the different type of the genome. So the plant genome are generally used for the analysis various purposes. There are several bioinformatics tools that can be used for the plant genome analysis. You can see here the assemblers, uh, APSIS, Velvet, like this. These are software tools are available for the plant genome analysis. Here are the URLs for the plant genome analysis. Plant genome database tools. We can use this type of the different type of the database tools. Bio extract server, MUSEC docs. These are the different type of the servers that can be used for the plant genome analysis. Genome databases are also available. There are several databases available online. Millet genes, rice genes, soil genes, sorghum genes database, soy genes also available on the internet. So plant databases are also available that are related to the genome. So plant genome database, white genome data, phytosomes, and legome information system. These information systems are available on. So there are several plant genome already completed genomic information, genomic plant genome structurally, and that are useful for the every type of work in the plant biotechnology. Similarly, the plant databases, plant genomic database are available. You can see this database you can use for the your plant biotechnology purpose. So again, there is a very important technique which is used in the bioinformatics, that is biodiversity informatics. Using the biodiversity informatics, you can uh, we can identify online interactive identification techniques for the plants. We can develop the online interactive identification of the plant. We can develop a digitized inventory of the plants, uses of the primary species, information system for the flora. So bioinformatics tools are responsible for development of this type of the work. So digitally enabled curation, you can see here that image of the digitally enabled. So plant distribution analysis also can be done by using the bioinformatics software. Thank you. This is about the role of the bioinformatics in a plant science. Thank you. Thank you all.